Assalamualaikum and greeting everyone. Welcome to my final year project presentation. I am Dalina Suhaili Binti Zakaria, a final year student in Bachelor of Science Honor Biology. The title of my final year project is Diversity of Macrofungal on Sandy Beaches in BACHOK, KELANTAN. I am supervised by Dr. Nur Azli Zabinti Ismail. This presentation consists of 5 chapters which are Introduction, Literature Review, Methodology, Result and Discussion, and also a conclusion and recommendation. Let's begin with Chapter 1, the Introductions. Fungi are crucial in maintaining the ecological balance of ecosystem. They help that root and leaf litter decompose and decay, allowing both simple and complex substances to be recycled. The fungi can be divided into 3 fundamental ecological types, which are mycorrhizal, parasitic, and saprophytic. In Malaysia, there is a lot of study that have been done on the diversity of terrestrial organisms on sandy beaches, such as trees and animals, but macrofungi are considered under-investigated. In addition, there were limited records of macrofungi diversity on sandy beaches in Malaysia. Furthermore, there is not enough knowledge of how macrofungi contribute towards maintaining the balance of beach ecosystem. The significance of this study are the documenting of fungi diversity and spread is essential for future reference due to insufficient studies on the variety and distributions of macrofungi in Malaysia. This research will also add updated knowledge of the diversity and distributions of macrofungi, particularly in Kelantan. Additionally, this study will provide more detail on their significance for maintaining ecosystem equilibrium. There were 3 objectives in this research, which are to report the diversity and distributions of macrofungi found on sandy beaches located in BACHOK, Kelantan, to observe the morphology of macrofungi using light microscope and scanning electron microscope, and to investigate the roles they play on the beach areas. Literature review will cover on several aspects. There are several characteristics that differ in macromorphology characteristics of macrofungi species, such as the cap structure, style, gills, vulva, and annulus. The micromorphology characteristic consists of basidia and basidia spores. Next, we jump into Chapter 3, Methodology. The study was conducted at Pantai Senok, Pantai Diaur, and Pantai Kemayan, which are located in BACHOK, Kelantan. The sampling process began with the specimen collections that collected during the rainy season from October 2022 until February 2023. Samples collected were labeled with collection number, date, and place. The samples were observed in site with taking pictures and taking the measurements of all specimens collected. The macromorphological structure, like the pillars, lamellae, and sheath, were observed and noted. The sample then taken to laboratory for microscopic observation using light microscope and scanning electron microscope. Only 70 macrofungal specimens were collected at the 3 sites in BACHOK: Pantai Diaur with 7 specimens, Pantai Senok 4 specimens, and Pantai Kemayan 6 specimens. With respect to macrofungal species, a total of 10 species were successfully identified, which were Pycnophora sanguinis, Habeloma species, Stera ammophila, Ganoderma species 1, Ganoderma species 2, Pleurotus species, Trimethys cubensis, Lentinus species, Trimethys species, Trimethys pubescens. Only 1 species could not be identified. 3 species, which is Pycnophora sanguinis, Habeloma species, and Ammophila, were recovered at all sites. The dominating family with the majority of diverse species documented in this study was Polyporaceae, with the total of 5 species, followed by Ganodermaticae, which were 2 species. Family Hymenogastraceae, Pisteraceae, and Pleurotaceae only have 1 species each. Polyporokeae can break down and decompose dead material. Dead organic material such as driftwood and fallen branches is one of the major reasons for their dominance in coastal ecosystem. Because of their effective lignin-degrading system, they can digest difficult woody substrate, promoting nutrient cycling in sandy coastal habitats. There were two types of macrofungi recovered, which were saprobic and macrohizal. Macrohizal macrofungi create mutualistic connections with the roots of plants, especially trees. This symbiotic association help both the fungus and the host plant. In these research, the macrohizal macrofungi was Hebeloma species. Saprobic macrofungi, on the other hand, degrade dead organic matter and play an important role in nutrient cycling and organic matter decompositions. They are usually found on decaying wood, leaf litter, and other organic material. The saprobic macrofungi in this study were Pycnophorus sanguineus, Viscerella ammophila, Ganoderma species 1, Ganoderma species 2, Pleurotus species, Trimethys species, Trimethys cubensis, Trimethys pubescens, Lentinus species, and unknown species. This species was suspected Pycnophorus sanguineus. The fruiting body is bright orange-red color. The pore reddish-orange in color. Cellular tube concolor to hymenophore. It ranges from tomentose to almost glabrous and possess concentric zonations. The spore was cylindrical, smooth, and transparent. Pycnophorus sanguineus are closely related to Pycnophorus cinnabarinus. Pycnophorus cinnabarinus has a vivid orange to red color, and the caps are often thicker and have a smooth or slightly wrinkled surface. This species was suspected Ganoderma species 1. The shape was irregularly kidney-shaped. The main color was brownish-red or reddish-brown, with or without few whiter zones. When young, the pore surface was white to pale brownish, turning medium brown with maturity. The shape of the spore was ellipsoidal to avoid double-walled with one end flattened. Some studies have highlighted the possible health advantages of Ganoderma. It has immunomodulatory properties, which means it can help control and improve immune system. This species was suspected Ganoderma species 2. The shape was irregularly kidney-shaped. The color was brownish-red or reddish-brown, with or without a few whiter zones. When young, the pore surface was white to pale brownish, turning medium brown with maturity. The shape of the spore was ellipsoidal to avoid double-walled with one end flattened. Ganoderma species have been found in Asia, in places like China, Japan, Korean, Indian, Malaysia, and Vietnam. They have a long history of usage in traditional Asian medicine and are highly valued for their therapeutic effects. The species was suspected Hebeloma species. The cap was tiny, fibrous, bell or convex with light ambonate, pale brown, the pillar surface smooth. The stipe shape was cylindrical to cleaved. Gills attached to the stipe, pale brown and crowded. The shape of the spore was ellipsoid, cylindrical, ovoid, amygdaloid, limoniform, fusoid, or even navicular. The species was suspected Viscerella ammophila. The cap was first bell-shaped, then flattened as it aged. The surface was smooth. Due to the old specimen, the lamellae were adnate and distantly packed. The lamellae of this species, on the other hand, are often dense and not distant. The gill's color was brown to chocolate brown. The Basidia spores were ellipsoidal in shape. Spores were smooth and having superciliar depression with a large germ pore. This is found mostly on the sandy coastline of the Atlantic Oceans, the North Sea, the Baltic Sea, and the Mediterranean Sea in Europe. This species was suspected Trametes cubensis. The shape was hemispheric to irregular. It also contains concentric zone that dominate radial sections. The pore surface was wide and irregular to angular in shapes. The basidia spore were ellipsoid or elongated in shape. Trametes cubensis were closely related to Trametes lepnia. Trametes cubensis is distinguished by an annual basidioma and small pore, whereas Trametes leptina has an annual to perennial basidioma and large pores. This species was suspected Trametes species. The shape was hemispheric to irregular. The cap surface contained a concentric zone. The pore surface is brown in color. The spore shape was cylindric to long ellipsoid and smooth. Trametes species have been found in a wide range of environments, from tropical rainforests to temperate woods. This species was suspected Trametes pubescens. The cap was semicellular, irregularly bracket shaped or kidney shaped. The cap was cream-colored with mild textural zones but no contrasting color zones. The pore surface was creamy and yellowish with age. Spore shape was smooth, cylindric in amylot. Trametes pubescens have anti-inflammatory characteristics that can aid in the reduction of inflammatory in the body. This species was suspected Lentinus species. The cap of this fungus is convex, then plain and funnel shaped. The gills are cream to dark cream, packed and decurrent. The spore print was white. The spore was cylindrical to narrowly ellipsoidal. Lentinus is a fungus genus that has a variety of species with varying ecological roles and possible benefits. This species was suspected Pleurotes species. The cap was fan-shaped or oyster-shaped with cream to yellowish color. The gills range from white to cream in color. Gills run down the stem. The spores are smooth and elongated. This species could not be identified. The shape of the cap was broadly conic to broadly bell-shaped. The gills were brown and attached to the stem. The base of the stem was swollen. The spore print was brown. There was no ring on the stems. The spores were mostly ellipsoidal with a prominent apiculus. This species possesses a similar morphology with the Agaricus species. However, it does not have a ring. The spore shape is also different than Agaricus species. The spores of Agaricus are often elliptical or nearly almond-shaped. Altogether, only 17 specimens were recovered at 3 beaches in BACHOK, Kelantan. 7 specimens were collected in Pantai Diau, 4 specimens were collected in Pantai Senok, and 6 specimens were collected in Pantai Kemayan. These researchers obtained a total of 10 recognized macrofungal species, with 1 species yet to be determined. The macrofungi obtained from this site fall into 2 groups, which were macrohizal and saprobic. For recommendation, comprehensive survey should be carried out to better understanding the diversity of macrofungi on the beaches of Kelantan. Molecular techniques such as DNA sequencing can be used to correctly identify and comprehend the taxonomy of the macrofungi. That's all from me. Thank you.